Welcome, welcome, and thank you for taking the time to view this CoLab presentation titled Structural Variation Detection with Genome Mapping and Human Chromosome Explorer. My name is Robert Lind, and I'm the Director of Product Management for the Life Informatics Center of Hitachi High-Tech. We are proud to be a silver sponsor at the 2020 ASHG conference. If you are viewing this CoLab during the conference, we encourage you to stop by our virtual booth or interact with our team at the virtual roundtable Friday, October 30th at noon Eastern. At any time, you can learn more about Hitachi High-Tech's Life Informatics Center and our projects by visiting structuralvariation.bio. Let's get started. In this presentation, you will be introduced to Hitachi's Life Informatics Center and the opportunity we see for genome mapping and structural variation analysis in both research and clinical applications. I will also demonstrate our whole genome map assembly and structural variation analysis pipeline called Human Chromosome Explorer. The Life Informatics Center is a virtual center that is part of a greater wave of small, nimble, and innovative project teams within Hitachi tasked with discovering, analyzing, and implementing technical and application solutions. Our mission is to develop data aggregation, integration, and visualization tools as part of a knowledge base for genomic life science research and molecular diagnostics. The team is at the beginning of this mission, and our first project is Human Chromosome Explorer, or HCE for short. HCE is an advanced web and cloud-based whole human genome assembly and SV analysis software for bionanogenomics and NABSIS genome mapping platforms. Hitachi strongly believes that genome mapping has the potential to advance genomic structural variation research and subsequently disrupt the cytogenetics landscape. If we look fundamentally and systematically at whole human genome structural variation analysis, we are at a state where sequencing is quite good at looking at single nucleotides in high complexity or multiplexing, and can routinely detect SNPs, small indels, and short tandem repeats. On the other end of the spectrum is karyotyping. This technique predates sequencing technology by many decades, yet still remains in standard practice today. It involves the cytogenetic practice of visually analyzing chromosomes under a light microscope. In addition to being used to ascertain chromosome counts, judge the position of centromeres, and note differences between the sex chromosomes, karyotyping can also identify banding patterns indicative of very large structural variation. It's the space in between sequencing and karyotyping that still represents a major technical challenge when analyzing structural variation in the human genome. And for good reason. Structural variation is multidimensional, spanning from unbalanced events such as insertions and deletions to balanced events such as inversions and translocations, to copy number variation, all the way to complex events such as chromoplexy, chromoanasynthesis, and chromothripsis. However, just having the ability to define different types of structural variation events doesn't mean that these events have been captured in their entirety, which of course means that these events have certainly not been fully translated. This isn't to say that structural variation is undetectable with current technologies. It certainly is. We've already mentioned karyotyping, which has remained in standard practice because it provides pathologic information that is observable for well-characterized genetic disorders caused by large structural variation. However, it's limited in specificity and cannot detect de novo or rare events. Further, the turnaround time due to culturing is slow, and it's expensive at around $11,000 per analysis. FISH improves upon karyotyping in that it is molecular and more cost-effective at about $1,500 per analysis. However, like karyotyping, it requires cell culture and is limited in multiplexing capability and throughput. Array CGH, like FISH, provides molecular information and is particularly good at detecting copy number variation. It's also relatively cost-effective at about $1,300 to $2,700 per analysis. However, the chromosomal locus is lost, which could be important for accurate pathogenesis, and it's purely prospective, eliminating its ability to detect previously unknown structural variation. In each of these cases, there is still a lack of diagnostic yield, suggesting that there remains much that is not being picked up in standard cytogenetic practice. The hope is that sequencing could fill in this gap and yield. However, Illumina short-read sequencing technology, while inexpensive and ubiquitous, cannot consistently resolve SV beyond 2 kb due to the fact that the sample prep protocol requires DNA molecules to be sheared to very short lengths in the hundreds of base pairs. Reassembling these very short reads to span breakpoints of large structural variation, and particularly repeated regions, has obvious inherent limitations. Also, despite advances in phase sequencing, the haplotype is often lost in the consensus. Capillary electrophoresis, while providing longer reads than Illumina and being particularly effective in the detection of short tandem repeats, still remains expensive with longer turnaround times and lower throughput. PacBio long-read sequencing technology has made great improvements in accuracy using circular consensus sequencing and can resolve complex SVs. However, the approach in many cases is limited to SVs up to 20 to 30 kb in length and remains expensive at lower throughput. Oxford Nanopore is another promising long-read sequencing platform that only recently started supporting SV calling. The platform promises read lengths in the hundreds of kilobases. However, typically reads fall into the 20 to 30 kb range, and the results are error-prone with rates ranging between 5 and 15%. Their latest flow cell and chemistry purportedly improve on this performance, but the approach relies heavily on consensus sequencing that may impact the haplotype. The takeaway here is that no single technology has proven to be a solution to the full complexity that structural variation in the human genome presents. This has led the research community to combine platforms to develop better references and improve SV detection. This lack of a single technology poses a fundamental problem when moving an assay into the clinic. If we think about the success Illumina or real-time PCR has had as both a research and diagnostic platform, it's not only that there was an assay that met a diagnostic need, but also the workflow, methods, and materials from the research setting could be replicated in the diagnostic setting. The requirement of combining technologies into a complex matrix of analysis may provide new insights into structural variation and is certainly critical to building better references, but the approach violates this fundamental rule for transition to the diagnostic setting. All of this points to an opportunity for genome mapping. This single molecule long-read approach to examining structural variation in the human genome is interesting in that it follows a familiar workflow, can detect de novo events, and offers the throughput and price performance needed to transition to the clinic. DNA molecules are enzymatically labeled and fluorescently stained in the case of optical mapping, or nicked and tagged in the case of electronic mapping, at sequence-specific sites. In the optical mapping approach, the resulting DNA molecules are linearized in microfluidic channels and imaged using high-resolution fluorescent microscopy. In electronic mapping, the linearized DNA molecules are passed through a nanodetector chip and the tags are electronically detected. In either case, the final outputs are single molecule maps, which record the sequence-specific label locations on each DNA molecule. The single molecule maps are assembled into map contigs. A reference sequence is then labeled in silico to generate a reference genome map. Structural variation can be identified by comparing the observed label pattern of the sample map contigs with the expected pattern of the reference genome map. For example, two label sites significantly farther apart on a sample map contig compared to the corresponding locations on the reference map could indicate an insertion. There are two commercially available genome mapping platforms: the Bionano Genomics Sapphire platform, which employs optical mapping, and the Napsys HD mapping platform, which uses electronic mapping. Single molecule map reads from either platform are uploaded to Hitachi's Hce, where they are assembled, aligned to a reference, and visualized as whole human genome analysis for structural variation. And while genome mapping lacks single base pair resolution, it routinely generates long reads, 50 kb to 300 kb and longer, that are necessary to resolve the most complex SV. This offers the potential of a single technology that can not only replicate the research and diagnostic utility of traditional cytogenetic platforms, but also provide the de novo analysis needed to close the gap in diagnostic yield. If we look at where genome mapping fits, it clearly fills the gap for a de novo molecular platform for structural variation detection and analysis. If we further consider the price per sample, it starts to become an interesting screening platform option, particularly now with the need for population-level analyses such as resolving misheritability, oncology studies, and other personalized medicine discoveries that require large cohorts and low costs. This could be similar to how microarray is used as a screening mechanism across populations. However, with genome mapping, de novo information is directly captured rather than inferred. Hitachi believes so strongly in the promise of genome mapping that we've developed a powerful secondary SV analysis pipeline to enhance the value of data generated from the Bionanogenomics and Hce platforms. In highlighting the key features of the analysis pipeline, we've gone back to the raw or pre-processed single molecule maps from Hce or Bionanogenomics to identify high-quality long reads as seeds. Map contigs are grown through a process of feeding these seeds through iterative comparisons of smaller reads throughout the dataset. It's in this iterative process that we enhance the single molecule map quality and automatically tune assembly parameters based on input data stats. This approach uses more of the available reads to create longer and ultimately fewer contigs in the final assembly. All of this is accomplished without seeding from a reference, which becomes significant as we move into SV analysis itself, since we want to accomplish de novo detection of SVs without the risk of a false negative due to reference bias. The reference map and aligned map contigs are annotated for any known genes. Identified SVs are checked against segmental duplication and gapped regions of the genome, assigned a haplotype, given a QC score, and have allelic fractions calculated. The resulting analysis is visualized in a web browser where further filtering and analysis can be done. Normally, this entire process would take more than 24 hours to complete, depending on the level of coverage and sample complexity. Hitachi has applied its expertise in cloud computing and large data management to dramatically decrease this entire process to 1 to 2 hours at 100x coverage. The key to HCE's speed and quality improvements is Hitachi's partnership with Google Cloud. Google Cloud, through its high-performance computing, or HPC, environment, allows us to dynamically spin up the optimal number of compute nodes to speed up iterative tasks and enable complex parallel processing of large genome mapping datasets. The results are high-quality de novo assemblies and accurate SV calls in dramatically shorter timelines, and without the introduction of compression, which could result in data loss. No discussion of cloud applications can exclude security. HCE is hosted in a highly secure environment we call the LIC security framework. This framework consists of a web application firewall on top of Google Cloud's advanced security features. Lastly, our partnership with Google Cloud allows us to deliver all the value described in a cost-effective service. The internal project for this workflow has been dubbed the monkey pod tree process, named after the Hitachi tree, which is a monkey pod tree around 130 years old that grows in Moanalua Gardens in Oahu, Hawaii. The name was adopted by the HCE development team because of how the assembly process uses the identification of high-quality long molecules as seeds to grow the contigs through a process of feeding these seeds through iterative comparisons of smaller molecules throughout the data set. The monkey pod tree process uses more of the available reads to create longer and ultimately fewer contigs. As stated previously, the assembly process was designed this way so as not to require seeding from the reference and preserve the integrity of the reads as purely representative of the sample, resulting in a truly de novo assembly. Let's take a look at the HCE platform itself. HCE has an intuitive user interface that was designed for all skill levels and is accessed through Google Chrome or Microsoft Edge. Raw data from Bionanogenomics or Hce platforms are uploaded with a few clicks. The uploaded data automatically undergoes a quality control check. The desired in silico reference genome is selected and the sample is submitted for analysis. The computationally intensive analysis pipeline is initiated and takes place entirely in the cloud. Single molecule read maps are error corrected and filtered. Assembly parameters are dynamically tuned. HCE's monkeypod tree assembly process builds high-quality map contigs which are aligned to the in silico reference, and whole genome SV analysis is performed. Typical samples take 1 to 2 hours to complete. In the results summary view, an overview of each chromosome is displayed showing genome map contig coverage of the reference and any SVs detected. Quickly zoom into regions of interest to view known gene annotations and information on any SVs called in that region. The results summary view also makes it simple to compare multiple samples, including the ability to hide SVs that are common among the selected samples to more easily identify differences in SV calls. Robust filtering options allow users to quickly navigate through results. Click on any region of a chromosome of interest to launch the alignment viewer. See how map contigs align to the reference at the label or tag level of resolution. The user can also toggle single molecule map depth information and mouse over any location or SV call to see more detailed information. Dive even further into the assembly by clicking on a region of interest in the alignment viewer. This launches the assembly viewer in which users can visualize depth and coverage of single molecule alignments at a particular region of a contig, allowing further validation of any SVs of interest. Finally, in addition to its visualization tools, HCE also has several built-in reports. Users can easily view and export input data, assembly, and SV stats. Sign up for your free HCE account and get full access to all the features of HCE on up to 3 samples at structuralvariation.bio. A deep understanding of structural variation in the human genome is needed to usher in the next era of diagnostic and therapeutic tools. However, the multidimensional nature of structural variation poses a significant challenge. While combining technologies can overcome some of the challenges in discovery, the approach does not have the reproducibility, ability to scale, and price performance needed to transition to the clinic. Genome mapping potentially offers long enough reads with the accuracy necessary for the next generation of SV research and diagnostics. Hitachi's Human Chromosome Explorer provides the informatics power at the speed and price performance required to realize the full value of the platform. We at the Life Informatics Center of Hitachi can envision genome mapping and HCE serving as a primary analysis and screening platform that is capable of advancing SV research and transitioning to the clinic. It is here at ASHG where cutting-edge research and cutting-edge technology intersect. Hitachi can help bring the technology, and we are looking to the community to bring the cutting-edge research. Thank you again for your time, and please visit structuralvariation.bio to learn more about the Life Informatics Center and Human Chromosome Explorer.